Let us have an introduction to biology. So, in this lesson, we're going to look at cytology, and then under cytology, we'll discuss the cell cycle. We're going to run through the types of cells, and then we'll talk about the cell organelles. So, to get started, let us understand what this cytology is. So cyto simply means cell, and then logy, now understand that it means study. So therefore, cytology, it is the study of cells. So when we're talking about cytology, it is all about cells, even these, they are cell organelles. Yeah, so this is what we'll be talking about. So now, proceeding, the definition for cytology, it is simply the study of cells and their features. That is cytology, the study of cells and their features. And then now, straight away, we're now going to discuss the cell theory. So this cell theory, it highlights what was discovered by scientists back in the day. So we're going to have a brief history, a brief background of the discoveries, and then we're going to summarize the cell theory and talk about it a little further. So the history behind the cell theory. So we'll start with the famous scientist Robert Hooke. So we have Robert. So this Robert Hooke, he's the one who experimented on cells. Yeah, so like he discovered that, he's the one who coined the term cells. Yeah, so where does this term come from when we talk about a cell? So it is because Robert Hooke, when he experimented using a microscope, when he was trying to see small organisms, like small things found in different things, like in plants, animals. So he saw things which looked like cells. So he saw tiny structures, very tiny fragments. So they're like, just, this is a cell. So he named them cells. Yeah, so he observed Koch cells. Yeah, so here on the history, we're just going to highlight the major things which were observed. So Robert Hooke did his work in around 1665. Yeah, so now let's quickly run through the other scientists who also contributed greatly to the cell theory. So starting with Robert Hooke himself. In 1665, Robert Hooke did his discovery. He described Koch cells and also other plant tissues. And then from there, we moved to the year 1673, where we had this scientist. He was from Dutch. So Antony van Leeuwenhoek, whatever this name is. So this scientist, he carried on with Robert Hooke's work. Yeah, he worked now with a more advanced microscope. Yeah, so how do you draw a microscope? Yeah, so that same thing. The microscope, this Antony van Leeuwenhoek, he worked on it, and then he discovered some cells in animal cells. So this he discovered cells such as the spermatozoa. So he just continued from observing the cells. So Robert Hooke he just observed that we have tiny cells, and then he called them cells in general. And then now Anthony continued from where Robert Hooke ended, and then he was able to see spermatozoa. He was able to see blood cells. So the blood cells, meaning he saw the red blood cells, the white blood cells. Yeah. So this was Anthony van Leeuwenhoek. Yeah. And then now we proceed to the other scientist from this one. So the year is now we're going to go to 1839. Yeah. So let us talk about Theodore Schwann and Matthias Sheldon. Yeah, so these guys they recognized that some cells are unicellular. So some cells are unicellular, while others are multicellular. So what this means, uni simply meaning that some cells they are made up of only one cell, one kind of cell, and then others like organisms rather. Some organisms are unicellular, others are multicellular, meaning they are made up of different kinds of cells. Yeah, so this was the work of these two scientists. So you can just take note. I've given a very brief summary so that you are familiar with where the cell theory is coming from. Yeah, so you can take note of these. They are common MCQs, multiple choice questions you can be asked. So in 1783, they tell you that Robert Hooke discovered something. Yeah, so they would want you to know which statement is wrong. Yeah, so this is just a quick summary for your studies. And then later on, after this, they discovered that cells are unicellular, cells are multicellular. We also had Rudolph. He was a German pathologist. So he specialized in diseases, pathology. So it was in the year around, this guy existed in around 1821 to 1902. So he continued with the work of Theodore Schwann and Matthias. Yeah, so this is around somewhere 1850s. That's when Rudolph did his work. Yeah, so this is where the study of cells originated, like different scientists. So you can even read more. There are a lot others. So, but these are just some of the major contributors. Yeah. So this is the study of cells. These guys were the pioneers. So now, after all these studies, after all these scientists did their study, together they form. There was now what was called the cell theory. So let us look at the summary of the cell theory. So this is our cell theory. So all these theories, they come from the discoveries of those scientists, others which we have not listed. So quickly, let's run through the cell theory. So if you're asked to write about the cell theory, you should be able to give this summary, which is having 4 things. So the cell theory, it is the fundamental unit of structure and function in living organisms. Yeah, so this is more of a definition of the cell. Yeah, so the cell theory, the fundamental unit of structure and function. Yeah, so this is the basic unit of a cell, or the basic unit of an organism. That's the cell. That's the cell. And then all living organisms are made up of one or more cells. So this is coming from the study of those two, Theodore and and the friend. So Theodor, Schwann and Matthias, these who have said, they said some cells are unicellular while others are multicellular. So it was included in the summary. So all living cells are made up of one, meaning unicellular, or more cells, meaning multicellular. And then all new cells arise from pre-existing cells. Okay, so this was another thing which was agreed upon. So like for us to have like this cell, it means it came from like another cell. So this cell can divide, we have two. These two cells again, they'll divide. So later on we're going to talk about genetics, we're going to talk about mitosis, meiosis, where we'll discuss about this in detail. So all new cells come from pre-existing cells, meaning all cells come from somewhere. Yeah, and then the other one, all biochemical processes are carried out by cells. Yeah, so all the processes like respiration, photosynthesis, all these biochemical processes, they are carried out by the cells. So this is the summary of the cell theory. Yeah, so you should be able to summarize it in 4 sentences. So now this same cell theory, however, it has some limitations. So let us quickly run through the limitations. Okay. So the limitations of the cell theory. So what is the shortcoming? So it can be asked, explain about the limitations of the cell theory or the shortcomings. Yeah, so this is what we're now going to talk about. So if we are able to know where the whole concept of cells is coming from, we should also be able to highlight some of the shortcomings. Yeah. So let us start with the first one. So we can talk about, you can just look at the cell theory itself and then you try to think of what's missing, like what can we understand from the cell theory. So the simplest one, it comes from here. So remember, these since these scientists said that all cells come from pre-existing cells, meaning that if we have a cell, it means it came from another cell. Also this cell, it came from another cell. It came from another cell, just like that. So but if this is the case, then where did the first cell come from? So where did the first cell come from? If all cells come from other cells, then where did the first cell come from? The cell theory doesn't explain this. So this is a shortcoming, this is a limitation. So where did the first cell come from? That is the first limitation. The cell theory does not explain this. And then the second limitation, so the second limitation is where we can talk about viruses. So the cell theory does not explain to us about viruses. It doesn't explain about viruses in detail. So the case is that a virus is considered to be a non-living thing. Yeah, so this is a virus. It is a non-living, we consider it a non-living thing because it doesn't satisfy all the 7 conditions for characteristics of living organisms. So if we're going to consider a virus a non-living thing, but how does the virus replicate? Like how does the virus reproduce? So should we consider it a living thing, a non-living thing? The cell theory couldn't explain that clearly to us. So this is another limitation. And then we can also talk about some complex organisms. So we can talk about complexity. So complexity, the cell theory, it does not address the complexity of multicellular organisms. So like it doesn't explain complex tissues. Like when we now go deep into the cells, when we talk about the different cells that entirely make up the human body, so there are some limitations. So in short, it doesn't explain the complexity. It will just tell you that no, all living organisms are made up of one or more cells, but it doesn't go very much further. Yeah, so these are some of the limitations to the cell theory. Yeah, so now we'll proceed to, so we've talked about the cell theory. So let us discuss the types of cells. So how many types of cells do we have? So we basically have two types of cells. We have prokaryotic cells and eukaryotic cells. So this is a picture showing a prokaryotic cell and a eukaryotic cell. So this is a prokaryotic and the other one a eukaryotic cell. So let us break it down step by step. So let's discuss. Firstly, where are they coming from? So remember, we're talking about the cell theory. So we said, according to Theodor Schwann and Matthias, they said some cells are unicellular. So some cells are unicellular, meaning some they can be made up of one kind of cell, and then others they are multicellular, meaning in some cells they can have a number of cells, different kinds of, so that's multicellular. So now this is where we now talk about prokaryotics and eukaryotics. So prokaryotics, they are unicellular, while the eukaryotic cells, they are multicellular. So no wonder we've now come to the types of cells, because we've learned about what a cell is and the theory which surrounds the cell. So now let us discuss these two cells, prokaryotic cells and eukaryotic cells. Alright, so now let's look at eukaryotic cells and prokaryotic cells and eukaryotic cells. So starting with prokaryotes. So if an organism is a prokaryotic cell, we call it a prokaryote. So these prokaryotic cells, like I said, they are unicellular. So we'll say prokaryotic cells, they are unicellular, simply meaning that they are made up of these cells which should be prokaryotic, will have most of them should unicellular, while the eukaryotic cells, they are multicellular. So simply meaning that they can be made up of two or more. Yeah, and then the eukaryotic cells, most of them, they are talking about the prokaryotic. So the prokaryotic cells, they lack membrane. So this is membrane. So they lack membrane bound organelles. So I'm going to talk about the cell organelles very soon from now. So the prokaryotic cells, they lack membrane bound organelles. So this will mean that obviously maybe these, they should have membrane they should have a membrane bound. They have membrane bound organelles. So what does this membrane bound organelle mean? When you say something is membrane bound, it simply means that a structure is having a cell membrane. Yeah, so like for example, in a prokaryotic cell, we're going to have structures. A structure doesn't have a membrane around itself. Yeah, but under eukaryotic cells, the cells which we find here. Oh, and then one major difference is that this one doesn't have a nucleus. So the prokaryotic cells, they lack nuclei. They don't have a nucleus. But for the eukaryotic cells, they have a nucleus. So this is a very easy difference to spot out. So here in the eukaryotic cells, it has a nucleus. So we're going to look at the structure of a nucleus. A nucleus has a membrane. We call it a nuclear membrane. And then inside of the eukaryotic cells, we also have mitochondria, the powerhouse. So this mitochondria, it also has its own membrane. Yeah, and then even some other cells which also other features of the cell, we're going to talk about them, which have a membrane. And also here we're also going to go we're going to list some cells which do not have a membrane. So here we're just giving a brief summary and overview of the differences. So in here they do not have membranes, but in here the cells which are found inside, they have membranes. Yeah, and then the eukaryotic cell, it is they are usually small. These, they are small, so like their length can be from 1 to 10 micrometer. Yeah, so they are very small. So remember, a cell is a very tiny thing. Yeah, and then we're saying the prokaryotic cells, they are even smaller as compared to the eukaryotic cells. So for the eukaryotic cells, they are around 10 to somewhere 100 micrometer. So even these, they are small, but at least they are larger compared to the prokaryotic cells. Yeah, so this is another difference, 10 to 100 micrometers. That is the range of size of eukaryotic cells. Yeah, and then now I'm going to show you a summary which talks about more differences. Yeah, so this can come as an essay question, like as simple as it is. Write the differences between prokaryotic cells and eukaryotic cells. So you should be able to say this one doesn't have a nucleus, while eukaryotic cells have a nucleus. Prokaryotic cells are unicellular, eukaryotic cells are multicellular. Prokaryotic cells lack membrane-bound organelles. So the organelles, they are simply parts of a cell. Yeah, we're going to talk about them. These small cells found inside. So most of them in the prokaryotic cells, they don't have a membrane, but in eukaryotic cells they have a membrane. Yeah, so and then the prokaryotic cells are smaller while eukaryotic cells are larger. So these are some of the differences. You should be able to list a lot of them, as many as you can. Yeah, so let me just display the summary which you can use as well as you study. So you can use this to study more about the differences between prokaryotic cells and eukaryotic cells. Yeah, so let us quickly run through. So some we have talked about them there on top, and then others they're just being mentioned here. So remember, this can be an essay question. It should be very easy, so you should be able to know a lot of them. The more points you give, the better your chances of getting higher marks. So let's quickly run through. We use the mnemonic DORA to talk about the differences. So we know this little explorer. So starting with DNA. DNA in prokaryotes it's naked, while in eukaryotes it's bound to proteins. So what this simply means, it is in this case whereby, like if this is a prokaryotic cell, this is a eukaryotic cell, the DNA which is found in prokaryotes, it is not membrane bound. Yeah, so remember these, they do not have a membrane, the cells found inside. And this prokaryotic cell, so this is a prokaryotic cell, this is a eukaryotic cell. So the prokaryotic cells, they do not even have a nucleus which has DNA. So like there is just something which is called the nucleoid. Yeah, we're going to talk about that in a few minutes from now. So they just have a nucleoid, so the DNA is naked. But here in eukaryotic cells, the DNA, it has a membrane, the nucleus has a membrane, and then it is bound to proteins. Yeah, and then in prokaryotic cells, DNA is circular, while in eukaryotic cells, DNA is linear. So the DNA in eukaryotic cells is in form of a line, but in here it is circular. It's because there is no membrane. Yeah, so like let me make this clear. So where can we write? Okay, let's just do this. So we'll say this is a prokaryotic cell. Prokaryotic cell. So in this cell, given that this is a prokaryotic cell, and then the other one is a eukaryotic cell. So then in the prokaryotic cell, the DNA, it is just circular. It is in a nucleoid. So a nucleoid, it is simply an area where we find DNA in prokaryotes. But in the eukaryotic cells, so this is a membrane. So it will have a membrane. And then the DNA in here, it is like we can say it is linear. Yeah, so later on, that is a topic which is way ahead, where we'll talk about the DNA double helix. Yeah, we'll talk about the DNA in its full length. So, but just to imagine what they're talking about, in eukaryotic cells, the DNA is linear. Yeah, but in here, it is coiled. But in here, it is linear. Yeah, so we're just giving a summary of all this. Yeah, so I think you have now gotten it. So in prokaryotes, DNA is circular. In eukaryotes, DNA is here circular, here linear. And then the other difference, prokaryotes, the DNA has no introns, while in eukaryotes, DNA has introns. So the introns, they are simply regions in a DNA. So like these introns, they do not code for amino acids. So remember, just having an introduction to biology. So all these, we're just looking at them on the surface. We're going to talk about them in full length. So explaining the introns. So in a DNA, like for example, this is a DNA molecule. So after some processes which take place, such as transcription, in a DNA molecule, you are going to find that there will be some regions. So there are some regions. So this is a region of intron, and then this part, maybe it's exon, and then also have other introns. So in DNA, For the prokaryotes, they do not have these introns, but in eukaryotic cells they have these introns. Yeah, so this we'll talk about it very nicely. We'll talk about the enzymes that removes introns, but that is a way, it is a topic which is ahead semester 2. We'll do that. So this is what you should know. I'm just trying to make sure that all is clear. Organelles, this will be the last topic we'll talk about. Organelles. So the organelles in prokaryotes, they have no nucleus. Remember, I said this is the major difference. In prokaryotes there is no nucleus, but in eukaryotes there is a nucleus. And then in prokaryotes there is no membrane around the organelle, but in eukaryotes they have a membrane bound. And then we're also going to talk about the ribosomes. So we have different types of ribosomes. So in prokaryotes we find 70 S ribosomes. Yeah, so 70 subunit ribosomes, and here we have 80 S ribosomes. Under reproduction, we have binary fission for prokaryotes, and eukaryotes have mitosis and meiosis. So just quick on this one, binary fission, we should be able to tell that it's whereby we have a cell and then it divides into two. So these two cells, they'll be exactly like the parent cell, but in eukaryotes some cells will reproduce through mitosis, where the cells will be similar to the parent, and then majority of them they'll reproduce through meiosis, whereby the daughter cells are different from the parent. Yeah, and then I'm even tired. Reproduction. Oh yeah, so still on reproduction, the chromosomes are single, while in here the chromosomes are paired. Yeah, so all these chromosomes, are going to talk about the chromosomes on genetics. The average size, so the ranges can differ. You just have to make sure that the value which you give is in the acceptable range. So for the prokaryotes, according to most textbooks, you'll find that their measurements was 1 to 5 or 1 to 10 micrometers. And then these, they are 10 going all the way up to 100 micrometers. Yeah, so that is on the types of cells. And then we can also quickly under the prokaryotes, remember the prokaryotes are unicellular. So now let's give examples of prokaryotes and eukaryotes. So we'll start with for the where do we start from? Okay, prokaryotes. Yeah, so under prokaryotes we have organisms such as a virus. Examples include bacteria. So we have bacteria. Under prokaryotes we also have archaea. So I don't know how you can pronounce this word. Yeah, so we can say archaea. So these are examples of prokaryotes, meaning these things, they are unicellular. It means they do not have a nucleus. It means their size is from 1 micrometer to 10 micrometers. Yeah, so these are the prokaryotes. And then examples of the eukaryotes, we have the protozoa. So we have the protozoans. We have the protozoans, and then we also have algae. So we have algae, although the spelling here it's AE. Yeah, so we have algae and we have fungi. So it means these, they are multicellular. Fungi. We also have plants. Now we also have plants and we have animals. So in secondary school, high school, we used to say the two types of cells are plant cells and animal cells. So plant cells and animal cells are actually, they are both eukaryotic cells. They are similar. So this is because they have different cells. Like the plant cells, they have different types of cells. Even the animal cells, they have different types of cells. No wonder we call eukaryotic cells, they are multicellular. Yeah. So these are examples. So common multiple choice questions you can be asked, like which of the following is an example of a prokaryotic cell? And then they'll give you so let's say you can be asked which of the following contains examples of prokaryotic cells, and then you are given A, they give you algae, algae, B, they give you fungi, and then C, they give you bacteria, and then D, they give you protozoans. Yeah, so here it is very easy to bacteria, it is an example of prokaryotic. Yeah, so the key to passing biology usually is to study everything. Make sure you understand. Yeah, so you can find that maybe in the lecture slides they do not include algae, and then here on the multiple choice now you see algae for the first time. You'll be like, I think algae is also a prokaryote. Yeah, so you should be able to read further. You can refer to the textbook and all that. So these are examples of prokaryotes and eukaryotes. So I also wanted to talk about the bacteria. We can have a quick review about the bacteria. So what does the bacteria look like? So the bacteria will look like this thing we're about to see here. So a bacteria, it is a prokaryote. So the DNA which we're able to see, it is like coiled. Sometimes it can be circular. So this is a prokaryotic cell of the bacteria. So you should be able to describe the bacteria. So you should be able to draw and label and give the functions. So let's quickly run through the functions of the bacteria. So number one, the capsule. So the capsule protects the bacterial wall. So we have this is the bacterial wall, like the outer layer, we can say the cell wall of the bacteria. It is protected by the capsule. Yeah, so that is the basic function. You can read more, but let's just give it a summary. It protects the wall, and then now this wall, it protects the inner parts. The wall protects the inner parts. And then the plasma membrane, this one which is in green, the plasma membrane, it controls what enters or leaves the cell. So we're actually going to talk about this as a topic. It's the next topic after this lesson. So the plasma membrane to control what will go in and what will go out. And then the cytoplasm, it is where the biochemical processes take place. Respiration, replication, yeah, all these processes will take place like in the cytoplasm. And then ribosomes, they are for protein synthesis. These ribosomes are the ones which synthesizes proteins. And then we can talk about the plasmid. So we have this plasmid. So the plasmid, it simply carries extra genes. Yeah, so it carries extra genes, and then, but this is not the major function. So it carries extra genes, and then now these extra genes, how do they help the bacteria? So they help the bacteria because they are antibiotic. They are antibiotic resistant. So this means like if you have like bacteria, oh by the way, when we say bacteria, okay, so bacteria, it is a plural, and then this cell, this thing was seen, it is a bacterium. So this is a bacterium, that is the singular. So the bacteria, what? The bacteria, or when you have bacteria in your body, yeah, when you are infested with bacteria, this plasmid, it is antibiotic resistant, meaning like you can maybe take in antibiotics, you are trying to kill it, but it also has a defense mechanism. The plasmid, it will be destroying the antibiotics you are taking. Yeah, and then we can talk about the villi. So this is supposed to be pronounced, spelled as villi, like that. So these, they are for attachment to surfaces. So for the bacteria to make sure that it hugs you, it gives you a hug, it will be by the help of the villi. So the bacteria, these, they are usually like germs. So like you can have maybe on a door, this is the hand of a door, if there are bacteria here, the bacteria will be attached to your hand through the villi, or we can say villi. Yeah, so that is on that. And then which other function do we have? So the nucleoid, it is a circular DNA, it is the part which has DNA. Yeah, and then the bacterial flagellum. So this one, it is more like something we are more familiar with. How do you draw a sperm cell? Okay, the drawing has gone. Yeah, so but we're familiar with a sperm cell. So a sperm cell has a head, a body, and a tail. So this tail, it is like the flagellum of a bacteria. So the purpose of tails, it is just to help in motility. Help in motility. What does it mean when we say motility? It means movement. So the bacterial flagellum, it helps in movement. Just like the tail of a sperm, it helps in movement. Yeah. So this is about the bacteria. So now we're almost done with our lesson. So now can we now rush to organelles? So this was supposed to be a live lesson, but network couldn't favor us. So if you have questions, you can just comment on the video. Yeah, while you have questions, you can just simply comment, and I'll be able to reply immediately. So let's talk about organelles. So this is a major part of this lesson. It is, well, I have a number of things to talk about. So let's break it down as easy as possible. Cell organelles. So there are a number of questions here. Usually multiple choice questions. Cell organelles. So what are cell organelles? Cell organelles, when I'm teaching this, I usually simplify it to say a cell organelle, it is a part of a cell. Yeah, this is the best definition I can give you. A part of a cell is a cell organelle. So let's understand better what I mean by part of a cell. So let's talk about this this human being. So if we say parts of a body, can you list the parts of a body? I'm sure you're able to say, okay, a body, the body of a person has a head, it has a hand. This is a part of a body. We have legs, the neck, yeah, whatever you're able to like see. So these are the parts of a body. So we can apply this to the cell. So when we talk about the cell, we're simply saying what parts are there when we observe it nicely under a microscope. So we'll be able to see that, oh, under a cell we have a cell wall, we have a cell membrane, we have a cytoplasm, we have a nucleus, we have mitochondria, we have endoplasmic reticulum. Yeah, these things, the parts of a cell are what we are calling cell organelles. Yeah. So now we're going to have a run through these organelles, and then before we proceed, remember we listed plant cells, and animal cells. Animal cells. So these are examples of eukaryotic cells. So now you should be able to give differences, like, okay, a plant cell has a cell wall, a plant cell has chloroplast. Yeah, so I don't plan on going that length. So you can just revise through this picture, which is very, very detailed. Yeah, so let's drop it down. So you can try and screenshot, or you can inbox me on WhatsApp for the notes. So the plant cell and animal cell. So this is a very microscopic picture giving you the cells, the parts of a cell. Yeah, so you should be able to spot out some differences. Okay, so here it has this one, it usually has a fixed shape, like it can, a plant cell, or this plant cell can have a rectangular shape. This one, it can have a rectangular shape, sometimes a hexagon. Yeah, whatever shape, like it will be fixed, it usually doesn't change. The shape is regular for the plant cell, but for the animal cell it's not. Yeah, so I don't indulge on going into much detail. Yeah, so you can just read further. This is an assignment. Yeah, so now let's continue with our journey on cell organelles. Yeah, so I think the introduction is clear, but in case you are not clear, you can just comment there where we talked about this. What about if it's like that? Yeah, you can bring out your ideas so that we add on to what has been discussed. Yeah, so now cell organelles, what are we going to talk about? So this is a picture from the KNAB website. So these are cell organelles, simply parts of a cell, things you'll find in a cell. The cell membrane, endoplasmic reticulum, ribosome, nucleus, nucleolus, all these, and this is what we're going to talk about in detail, or not in detail, but individually. So let's get started. We'll start with the nucleus. So a magnified nucleus will look like this. So when we're explaining about a cell, we simply just say, okay, cell has a nucleus, but this nucleus, it has more than we would just draw. So a nucleus, it has a membrane. Always remember, eukaryotic cells have a membrane. So a nucleus has a membrane, and that membrane, we call it the nuclear. We call it the nuclear membrane. The membrane around the nucleus, we call it the nuclear membrane, which can also be called this nuclear envelope. Yeah, and then now on this same nucleus, it's going to have pores, some tiny pores through which nutrients can pass through. We call those nuclear pores. Nuclear pores, tiny openings. So you can see these. So these are the nuclear pores. Yeah, and then what else can we talk about? It has a nucleolus. So the nucleolus, it is the actual area now where we get to have the DNA. And then we also have chromatin. These later on, when they like mature, when they come together, they form a chromosome. The chromatin, yeah, it is found in the nucleolus, and it gives rise to a chromosome. Yeah, so this is what we can say about the nucleus. Simple and straightforward. Yeah, so let's proceed to the next. So my target is to finish this lesson before an hour. So now next we can talk about the we have this area which is called, so we're still on the DNA, so like we're just talking about the DNA. So remember, in prokaryotes and we have eukaryotes. So in prokaryotes there's no nucleus, and I said that instead of a nucleus we have a nucleoid. This, it is an area which will have a DNA. Yeah, and then but in here we have a nucleus. So where is the nucleus here? It is this thing. But in prokaryotes there's no nucleus. And then by now you have understood very well the function of a flagellum. It is for motility. Motility means it helps in movement. Yeah, so that's on the DNA. And then what else can we discuss? We can talk about the this ribosomes. So we're going to talk about the ribosomes. So by this picture you're able to see, it is a picture of a mitochondria. So a cell will have mitochondria. So this is our cell, and then it has a nucleus, and then it's also going to have a powerhouse, a mitochondria. Yeah, so but this is not drawn to scale. The way I've drawn it doesn't mean that the mitochondria is larger than the nucleus. The nucleus, it is actually the largest. It is larger than the mitochondria and all other things. So in the mitochondria, so let us even talk about the mitochondria. So the mitochondria, it is the powerhouse of the cell. It plays a big role in respiration, the production of energy. And then the structure of the mitochondria. So the mitochondria, it is found in eukaryotic cells. And all organelles which are found in the eukaryotic cells, most of them rather, they have two membranes. So we say they are membrane bound. So like this is the mitochondria. So like this is our mitochondria, and then this same mitochondria, it's going to have its own membrane. This is what we say when we mean when we say membrane bound. Yeah, so we have the mitochondria. It has two layers. Yeah, and then there are also these structures inside. We have the ribosomes. So the ribosomes, they are there for protein synthesis. So actually the mitochondria can also be on the limitations of the cell theory. The cell theory does not explain how the mitochondria has its own nucleus. So it has its own DNA. The mitochondria has its own DNA, meaning that it can reproduce. The cell theory does not explain that. So it has DNA, it has cristae. These are the four genes of the mitochondria. And then we have this space. So we're just basically summarizing this structure. Yeah, we don't go into much detail. Yeah, so but can we quickly just run through? Okay, yeah. So where can we start from? Okay, yeah, so we have the mitochondria as an outer membrane. So the function of the outer membrane, it simply regulates the movement of materials in and out of the cell. Like it will regulate the movement. And then as for the inner, that's the outer membrane. And then the inner membrane, it simply permits what to enter and what to leave. So this one, it's just a passage, and it will protect the inner parts. And then the inner membrane, this membrane. So this is the inner membrane. It regulates what enters and leaves. Yeah, and then we have other parts like the matrix. So the matrix, it is just a space where some processes take place, such as the Krebs cycle. So the Krebs cycle, it is not for your level. Yeah, the Krebs cycle, it will be learnt by those who will be doing biochemistry and those who will be doing medicine and pharmacy. You look at the Krebs cycle, a very tiresome cycle. Yeah, and then we have the cristae, where the cristae here, they are the four genes. They simply increase surface area for respiration. Yeah, DNA, ribosomes, protein synthesis. Yeah, and then we can proceed. So what other cell organelle can we look at? So we can talk about so we can now go to like chloroplasts. So the chloroplasts they are predominant cells found in they are predominantly found in plant cells. Yeah, they play a big role in trapping sunlight. They contain chlorophyll so that photosynthesis will take place. Yeah, that is the basic idea. So you can read more about these. I won't go into much detail. Yeah. So, but we're just looking at the structures so that you see how they actually look, and yeah, you can have a broader picture. Yeah. So this is the chloroplast. We proceed. We can also talk about the endoplasmic reticulum. Which one should we go to? Okay, yeah, so we can talk about the ribosomes in like more detail. So this is the structure of the ribosome. So here the ribosomes, they are very tiny things. Like this is the cell. The ribosome will look like a dot, just like a dot. But when we magnify it, it will look like this. So a ribosome has two parts. It has a large subunit and a small subunit. Yeah, so there are two types of ribosomes. We have the 70S ribosome and the 80S ribosomes. So you should be able to know the 70S ribosomes, they have 50 and 30. Yeah, so we don't like just add them to get that. Yeah, so they are more complex stuff. Yeah, so you just have to take note that 70S ribosomes has a 50 and a 30, but the 80, it has a 60S subunit and a 40S subunit. So remember, it's not like this 8, it comes from adding this. Yeah, so we're going to discuss more about that later on. So like the functions, they are simply for protein synthesis, but they have even more functions such as translation of mRNA. Yeah, we're going to talk about transcription later on. Yeah, so but for now I think we can just take it that it is for protein synthesis, and then it is also, both of them, they're also used for peptide bond formation. Yeah, the key difference is that for the 70S, they are found in prokaryotes, 80S, they are found in eukaryotes, and the 80S subunits, they are more complex. Yeah, and then we proceed. Yeah, so I haven't managed to reach my target. I wanted this to be like within an hour. Now there's just so much to discuss, and then we can't skip some of these things. Yeah, so now we can talk about the endoplasmic reticulum. Yeah, so what should we know about the endoplasmic reticulum? So we have two types of endoplasmic reticulum. We have the rough endoplasmic reticulum and the smooth endoplasmic reticulum. So the difference comes in, so like let's say this is our endoplasmic reticulum. This is our endoplasmic reticulum also. So they are just similar, the endoplasmic reticulum. The difference is that the rough endoplasmic reticulum, it appears rough because it has ribosomes. It has ribosomes. So the rough endoplasmic reticulum has ribosomes. So there are some endoplasmic reticulums which do not have ribosomes, and then others have ribosomes. These which have ribosomes, they play a big role in processing of the synthesis of proteins. Yeah, so but let's quickly talk about the functions of each. So starting with the rough endoplasmic reticulum, the function, we can start with the synthesis of secretory proteins. Yeah, so like the main function, it's all about proteins because it has ribosomes. Yeah, and then you can also look for other functions. So number one, you should just always remember to give this function. It involves in the synthesis of secretory. So secretory proteins simply meaning that these are proteins which will enable how can I put it? Like these will be proteins which will aid in like secretion, the removal of some unuseful substances. Yeah, so like it is useful in secretory proteins. These are the rough endoplasmic reticulum. So secretory proteins such as hormones and enzymes. Yeah, and then the major function now, it is about processing and packaging. So like remember, ribosomes, they are the ones which synthesize proteins. These ribosomes are the ones which will like make the proteins. So when the proteins are made, they'll be packaged by the rough endoplasmic reticulum. Yeah, and then after being packaged, they'll even be transported. So this is again another function. So the ribosomes will make proteins, and then the rough endoplasmic reticulum will pack them, and then it will transport them to where? To the Golgi body. So this is an organelle which we are going to talk about. Golgi apparatus or Golgi body. And then for the smooth endoplasmic reticulum, these they do not have ribosomes, so they don't play much role in things related to proteins. Yeah, so like here, I'm not sure if I've made myself clear, but you can comment so that in case you would want an explanation somewhere. Yeah, so but secretory synthesis, yeah, they play a big role in that. Uh, they play a big role in the synthesis of enzymes and hormones. Yeah, so now going to the smooth endoplasmic reticulum. So these their main importance is in steroid cells. Yeah, so and also like things like the liver cells. So their functions, when you just hear the liver, then they deal with toxic things like toxic substances. They'll be dealt by the smooth endoplasmic reticulum. Yeah, so like if someone drinks like alcohol or drugs, the liver, it will have abundant smooth endoplasmic reticulum so that it can work against those. Yeah, then it also synthesizes lipids and cholesterol and other things. Yeah, I think this is what we can talk about these. So for the diagram, what can you refer to? Yeah, so you can make use of your textbooks so that you have a clear picture. Yeah, so but this is just a simple diagram. The major difference is that when you just see ribosomes, then this is a rough, and then here there are no ribosomes. This is a smooth endoplasmic reticulum. So remember, on the rough endoplasmic reticulum, it has ribosomes. So now these ribosomes, they produce enzymes, and not producing, like they synthesize, they make enzymes. These enzymes, they'll be packaged by the rough endoplasmic reticulum, and then the rough endoplasmic reticulum will take the proteins to the Golgi body. So now let's talk about the Golgi apparatus. So the Golgi apparatus, I think I have a picture for this. So the Golgi body or Golgi apparatus, it looks like something like this. So it works hand in hand with the rough endoplasmic reticulum. So that thing in blue you can see, it is the rough endoplasmic reticulum, and it has ribosomes. So when ribosomes now they work on the proteins will come to the Golgi body or Golgi apparatus. And then now what will the Golgi apparatus do? The Golgi apparatus will now pack. It will continue with the packaging. So it will pack these enzymes, which were also packed by the endoplasmic reticulum. Yeah, but maybe with a few modification, and then it will sort them out. It will sort them out like, okay, this protein should go to the liver, this protein should go to the brain, this protein should go to the intestines. It will sort them out, and then the packaging starts. And then through the same cell, like you can see here, this is a plasma membrane. Like maybe this is, like we can say, a protein. It is here, it has been sorted, and then it is released. Yeah, so you just have to keep it simple. You don't have to write a full page to show that you've understood. With just a few pages, as long as you've gone straight to the point, you'll be able to get the marks required of you. So we can talk about this other cell organelle, which we did not list, but it's very important. So these are the lysosomes. So the lysosomes, they are the killer cells. I can say that they are the white blood cells of the cell. So what does this mean? It simply means that their function, it is mainly in how can we put it? Okay, so the lysosomes, they get rid of cells which are not needed in the body or in an organism. So like we have a lipid bilayer, so I'm not interested in the parts of the lysosome. So take note, we're looking at organelles. So under organelles, we have reached lysosomes. So again, these lysosomes, I've written lysosomes. Yeah, okay, so it's supposed to be lysosomes. So these lysosomes, they also have their own organelles, which are the lipid bilayer, which will just protect the things, which will select the things which will go in and out. And then inside it has hydrolytic enzymes. Yeah. So now the functions of the lysosomes, it's simply to get rid of substances which are not needed in the body or in an organism. So, and how does it do it? Or we can say, what are the major functions? Like, so the first one, it is intracellular digestion. We have intracellular digestion. So we have intracellular digestion. It is whereby, like, maybe you eat something, this is your mouth with your head like a chicken, and then this is your stomach. Hey. Okay, so this is the esophagus, and then we have the stomach like that. So this is a human being, very fat. So now we have something like this. So when you eat something, like you eat, so now there will be things which will not be needed, like maybe it can be food substances or just some other substances. So these things which will not be needed in the body, they'll be gotten rid of by the enzymes. So this is now what we call intracellular digestion. This is whereby the lysosomes, so like this lysosome, it will now go to the food. It encloses it, or like the membranes will fuse, like it will be next to it, and then it will start releasing hydrolytic enzymes. When it releases them, then it will kill these, and then they'll be ready for excretion. Yeah, so that is one way of the action of lysosomes. And then the other one, we can talk about autolytic action. So what do you have to know on autolytic action? So there are different ways in which this can be explained, and one of them is like just through this example. So here, a better explanation is whereby the lysosome, it will like get involved with what has to be gotten rid of, and then it directly releases the hydrolytic enzymes. Yeah, that is intracellular digestion. So it is very related to autolytic action, because autolytic action, it is now whereby like we have the lysosome, and then this lysosome, they'll be covering like this, want to get rid of this. So the lysosome, it will release the same hydrolytic enzymes so that we kill what we want to get rid of. So this is like, I can just say a different way of saying the same thing. Yeah. So, but if you are told to list, just list them, and then you can also do further reading. You can add more to that. So here I just came to give the summary. So there are some enzymes there are some organelles which I've actually skipped. Like we have peroxisomes. Peroxisomes, they are not much of focus, but you can just read them so that you are very familiar with any kind of question that can be brought towards you. Yeah. So, but the functions of these peroxisomes, they are simply organelles that have an enzyme which is called peroxidase. What's the name of the enzyme? I'm trying to remember. Peroxidase. Yeah. So they have an enzyme called peroxidase. Peroxidase. Yeah. So their function, it is mainly to catalyze amino acids and fatty acids. Yeah. So this is not very much of focus, so I couldn't even remember it very well. So the full spelling, it is peroxidase. Yeah. So remember, I said I've skipped this just so that I focus much on the things which you just have to, like, at least you should have on your fingertips. Yeah. So and the structure of the peroxisome, it will look like this. So yeah, so the peroxisome, so the peroxisome, it looks like this, which we have here. Yeah. So their function, it is mainly to oxidize fatty acids and amino acids. And then we're looking at the lysosomes. So the lysosomes, they get rid of the things which you do not need. They act by intracellular digestion and hydrolytic action. Yeah. So from here we can now go on to the last one. We can talk about the cytoskeleton. So the cell has actually a skeleton. So the skeleton, it is this membranous part. Yeah. So this is the skeleton. The function, I think, it is basically, you can just say, to hold the cell organelles in place. You can see we have the mitochondria, and then some other structures are not shown, but this is like the rough endoplasmic reticulum. Yeah. So these, they will be held together by the cytoskeleton. Yeah. So these are the cell organelles which you can take note of. Yeah, these you can't afford to forget. So there are a lot of functions of the cytoskeleton. We have to maintain the shape. It maintains the shape, and then it provides movement, motility. So it provides movement, and what else can we say? Through the cytoskeleton, it can help in transportation. Yeah. So the cytoskeleton, it has further structures like the intermediate filaments, the microtubules, the microfilaments, which are called actin. Yeah. So these, they are not much of my focus. Remember, just having an introduction to biology. You don't have to exhaust it as if you want to learn everything in one day. Yeah. So and for this, you are not going to focus much about them in your first year. Yeah, as you are starting your biology, you'll be able to learn about these mostly for medical programs. Those who will be doing medicine, you look at these in medical physiology, also pharmacists, biomedical scientists, and all that. Yeah, so I don't want to focus much on these. They have just been listed for you to know, like, okay, in the cytoskeleton, we have intermediate filaments, we have tubulin, which are microtubules, and all these. Yeah, so this is where our introductory lesson ends. Yeah, how I wish it was a live class. We could have a lot of questions, I'm sure. Yeah, so in case you have any question, you can just you can comment if at all you have any question, or if all is clear. Yeah, you can let me know, like, okay, I think I've gotten much of the concepts. Yeah, so we have achieved our objectives. Cell theory done, types of cells done, organelles done. Yeah, so our introduction to biology is complete. So I'm going to give you some work. Where's the work? So you are going to have this. So this is the tutorial sheet for biomolecules and cells. So your homework is to answer questions from question 1 up to 7. Yeah, so in the next lesson after this, we'll talk about the cell membrane. Yeah, so the cell membrane, it is, so if this is our cell, the cell membrane, it is just this thin line, but we're going to talk about it in more detail, where we'll say the cell membrane is a fluid mosaic. It is actually like thick. If we magnify this, it will look like that. Yeah, so homework, do question 1, 2, 3, 4, 5, 6, 7. Yeah, I want to see, or I should be able to get 100%, or at least above 80%. Yeah, so see what you can answer. I think I've explained much of this. You can even do some more research. So do this work and show me your answers, so that I see how you're going to write your essays. Some people get low marks because of not writing what is required. You are not starting well, your work is not well organized, and that will cost you. So you can inbox me your work on this WhatsApp line, and then I'll be able to attend to you. Yeah, so we'll end here. We'll continue later on. So just make sure to do this work. So you can screenshot and then answer the question 1 all the way up to 7. Yeah, and then also even send the questions in a group, so that you try them out. Yeah, so this is where we'll end with our biology introduction. See you in the next lesson.